In this talk, a paper recently published in Nature will be discussed. Hematopoietic loss of Y chromosome leads to cardiac fibrosis and heart failure mortality. Not only do men lose hair, muscle tone and knee cartilage as they age, they also begin to lose the Y chromosome in their cells. Scientists have linked the disappearance of the Y chromosome to a range of diseases and a higher risk of death, but there is still a lack of sufficient direct evidence. On July 14, researchers at the University of Virginia in the United States published a paper in Science that after removing the Y chromosome of male mice, these mice died earlier than those with Y chromosomes, which may be because the heart became more rigid. John Perry, a human geneticist at the University of Cambridge, UK, who was not involved in the study, believes this is the best evidence yet that losing the Y chromosome is harmful to health. The Y chromosome carries only 71 genes, less than 1/10 as many as the X chromosome, which may be why this chromosome sometimes fails to pass on when cells divide. Analyzing a blood sample is the easiest way to detect a Y chromosome deletion. The researchers found that in about 40% of 70-year-olds and 57% of 93-year-olds, at least some white blood cells were missing a chromosome, and in some older men, more than 80% of cells may be missing a Y chromosome. Cells can survive and reproduce without the Y chromosome, but men who lack the Y chromosome in certain cells are more prone to heart disease, cancer, Alzheimer's disease and other diseases associated with aging. University of Virginia molecular biologist Kenneth Walsh, one of the corresponding authors of the paper, said this may be one reason why men in the U.S. live on average 5 years shorter than women. To test whether removing the Y chromosome would harm health, Walsh's team performed bone marrow transplants on 38 mice. They used the CRISPR-Cas9 gene editing tool to delete the Y chromosome in mouse bone marrow cells, then inserted the altered cells into young male mice whose bone marrow had been removed. Instead of removing the Y chromosome from the recipient, the replacement adjusted the proportion of Y chromosomes in white blood cells to about the same proportion as many people with Y chromosome deletions. 37 control mice also received bone marrow transplants but retained their Y chromosomes. The researchers followed two groups of rodents for nearly 2 years and found that mice lacking the Y chromosome were more likely to die during this period, with only about 40% of them surviving 600 days after transplantation, compared with mice in the control group. About 60% survived 600 days. The hearts of mice that lost their Y chromosomes also had weaker hearts. About 15 months later, the strength of the heart's contractions decreased by nearly 20%. In addition, in the hearts of mice lacking the Y chromosome, tough connective tissue builds up, a process called fibrosis proliferates. This buildup can harden the heart, impairing its ability to pump blood. The researchers' bone marrow transplants in mice did not eliminate the Y chromosome in the heart muscle cells, but the macrophages produced in the bone marrow slipped into the heart. The scientists found that many macrophages in mice lacking the Y chromosome started to promote fibrosis, stimulating other cells in the heart to develop more connective tissue. A similar situation could happen in humans, and Walsh's team obtained DNA and survival information on more than 15,000 men from the UK Biobank. They found that men with a 40% deletion of the Y chromosome in their white blood cells were 31% more likely to die from circulatory diseases than men with more abundant chromosomes. When the researchers further analyzed the causes of death associated with Y chromosome deletions, they found that several heart diseases, including heart failure, were closely linked. Losing the Y chromosome stimulates cardiac fibrosis, leading to heart failure and earlier death, says Walsh. Researchers have long underestimated the health impact of the Y chromosome because it carries few genes, but there is evidence that its absence can dramatically shorten lifespan. However, the results do not confirm that the increase in fibrosis, which is fairly subtle in animals lacking the Y chromosome, kills the mice, cautions Nikolaos Frangogiannis, a cardiologist at the Albert Einstein College of Medicine. A weakened heart in rodents is not that bad or fatal, and the mice may have died from some other heart-related cause. Still, he agrees that the research is very exciting and could have a major impact on the way heart failure is understood. So we have completed the introduction of this paper. Thank you for your listening.